All of us are plagued by the big questions. So why do we do the things we do? Where does animal behavior come from? Why do some behaviors seem learned, while others seem to be encoded into our genes? This question has fascinated Cornelia Bargmann ever since she was a young girl.
How is it that something as fixed as a gene can give rise to something as complex and variable and flexible as a behavior?
It's one of the biggest mysteries in biological science. How do genes influence decisions? Early in her career, in pursuit of answers, Corey Bargmann developed her trademark research philosophy. For big revelations, look to the smallest of places.
C. elegans, a simple roundworm found in compost heaps, in soil, and even under apple trees. And it has only 302 neurons, compared to the 86 billion found in our own brain.
For scientists, a simple organism is easier to study than a more complicated one.
And I looked at this map of all of the neurons and all of their connections, and I said, this is the animal for me.
Somewhere within an organism no bigger than a comma on a printed page lie the connection between genes and behavior, and Bargmann set out to uncover it. She would start by studying a primal instinctive behavior common in most animals, the tendency to gravitate towards things that smell good. And Bargmann knew that one odor, a chemical called diacetyl, was a C. elegans favorite. They can't get enough of it. It was a predictable behavior, and Bargmann wanted to know what caused it. To make progress, she'd first need to locate the odor receptor responsible for sensing the chemical. And to do this, she created mutant worms.
You start with a large number of genetically identical animals, and you treat them with something that causes mutations in their DNA. And we asked them whether they liked this particular chemical, diacetyl. 9,999 of the worms went to diacetyl, which is what worms are supposed to do. And one of those worms said, "Did you ask me something?" And it didn't go anywhere.
The mutant worm lacked a receptor that the other worms had, a tiny odor receptor. And if she could find the genetic mutation responsible, she would know exactly which gene was required for sensing the odor. Bargmann spent long nights poring through the code by hand.
So I was reading the code by hand, going through this gene, because we'd been working on this for so long. And I realized, this is the odorant receptor, this is the receptor for diacetyl, we have it.
Bargmann had done it, but perhaps the most fascinating aspect of her research had just begun.
Olfactory or smell receptors are affixed to neurons like this one. After locating the receptor for diacetyl, a scent the worms like, Bargmann's team found on a neighboring neuron a receptor for scent the worms didn't like, a chemical called nonanone. This raised an interesting question.
What is it that determines whether the odor is attractive or repulsive? Is it the receptor? Does the receptor actually carry that information? Or is it the neuron?
Bargmann and graduate student Emily Tremmel used a series of techniques to create mutant worms with good smell receptors on the neurons that initially hosted the bad smell receptors, and vice versa. Bargmann and Tremmel then exposed the worms to diacetyl, the good smell that typically causes an attractive response. What Bargmann saw was remarkable.
Instead of being attracted by it, they were appalled by it. They ran away from it as quickly as they possibly could. They viewed it as a dangerous odor.
The key here is neurons determine behavior. One day, using techniques similar to the ones developed by Bargmann, scientists may better understand the biology of unhealthy behaviors. Ultimately, though, Bargmann's findings provide some of the deepest insight yet into why animals behave the way they do. They make Bargmann one of the first people to ever fully describe the behavior of an organism from start to finish, from genetic roots through the activation of a neuron to the behavior it produces. In other words, Bargmann shows us how a gene leads to a choice. And this is big news, not only for biology, but for all of science.